So this lab. So for this lab, I'll be doing the extraction of DNA from food. All living cells contain genetic material in the form of chemical DNA, or in the case of some viruses, they have RNA instead. DNA determines the characteristics of an organism, such as their behavior and their form and so on. So DNA is made up of nucleotides. Nucleotides are repeating units. They are made up of phosphate group, a sugar group, and a nitrogen base. There are four nitrogenous bases: adenine, guanine, cytosine, and thymine. The ability to extract DNA is one of the primary importance of studying the genetic causes of disease and for the development of diagnostics and drugs. It is also essential for carrying out forensic science, sequencing genomes, detecting bacteria and viruses in the environment, and for determining paternity. As the first step, I made a lysis buffer. The lysis buffer lyses the nuclear membrane as well as the cell membrane. It separates DNA from other cell debris. In this experiment, I made the home-based lysis buffer consisting of 500 ml dish detergent, 15 g of salt, and 950 ml water. The salt neutralizes the negative charge of the negatively charged phosphate in DNA. The detergent helps to solubilize the membrane protein and to rupture the cell membrane to release its contents. To extract DNA for this activity, it is best to use mushy fruits such as banana, and I used 2 bananas in this experiment. The skin was peeled off and placed in a Ziploc bag to mash it until it becomes a juicy mush. Mashing the banana exposes a greater surface area from which to extract the DNA. It was made sure there were no banana clumps in there. About 10 ml of the prepared lysis buffer added in the mushy banana soup. I used a strainer with some tissue to extract the solution. After waiting for about 10 minutes, there was some thin layer of solution. Then I added some ice-cold isopropyl alcohol, which is the hand sanitizer containing 70% alcohol. The addition of ice-cold ethanol slowed down the enzyme. The ethanol also served to promote the aggregation of the DNA. I waited for a few minutes to get the gel-like substance. The cloudy gel-like substance is the DNA of banana that can be picked up using a wooden stick. The salt used help break down the cell wall causes the DNA to clump together. The salted DNA is insoluble that begin to precipitate out of the mixed banana soup. It transforms from a dissolved solute into soluble substance in the ethanol layer.